In this video, we will be talking about how proteins are made, how they're coded for, tRNA, which is very difficult for me to pronounce, central dogma, etc. So let's get right into it. First of all, when we talk about central dogma, it's essentially the steps that a protein undergoes to become encoded, right? So it's the steps that DNA takes. So first of all, in the nucleus, we have the DNA, obviously. But to make protein from DNA, the DNA has to undergo transcription. So what is transcription? Transcription is when you go, let's put it over here. Transcription is when you go from DNA to RNA, right? And then after we go from transcription, we have to go into translation. So translation is when you go from RNA to protein. It's very exciting, I know. So again, transcription, it just applies in the nucleus, and translation is often in the cytoplasm. So this is typically in the cytoplasm. Right? So what's the difference between RNA and DNA? Well, in DNA, remember that we do not have an alcohol on the second carbon. So on the first carbon, we have an alcohol, but on the second carbon, we don't. That's just a hydrogen. So we call it a deoxyribose. So for instance, DNA stands for deoxyribose nucleotide, right? So, yeah. Deoxyribose nucleic acid, sorry. So for ribose, right here, it's used in RNA, and notice that in RNA you do have an alcohol. So RNA has, let's say, second alcohol, okay? Right there. And one of the major differences that you'll always see on biology exams is that instead of thymine, you use uracil. So it's not that difficult to remember uracil. Uracil is actually classified as a pyrimidine, right? So it's a pyrimidine, meaning that it only has one ring. A purine, which is adenine and guanine, have two rings. And a similarity between thymine and uracil is that uracil does not have the methyl group. So notice that thymine has a methyl group, which is a CH group, CH3 group, and uracil does not have that. So uracil has no methyl group. Other than that, it's pretty similar to thymine. So typically on biology exams, they'll say a question like, if you were to replace one nucleotide for this nucleotide, would the function change? And you would say, well, not really. Thymine can be replaced with uracil, and it wouldn't really change, you know, the structure. They're very similar. So as you can see that here, uracil is binding with adenine. So uracil goes with adenine within RNA. And it has two hydrogen bonds. So we have two H bonds. So is this connection strong? No. The only strong connection would be between guanine and cytosine, which have three hydrogen bonds. For uracil, adenine, or thymine with adenine, those are pretty weak because they only have two hydrogen bonds. Now, we look at the structure of RNA. So if I were to see this in a textbook, I would say, yeah, this is RNA because we have cytosine, adenine, but here we have uracil. And then here we have guanine. So just seeing uracil, I instinctively know, okay, this is RNA, right? This is ribose nucleic acid. And notice that, again, we still have a 5' to 3' polarity. So we go from the 5' to the 3'. And remember in the last video where I was saying, well, you can link them however you want. Notice that they have a cytosine connected to an adenine, adenine connected to a uracil, uracil connected to guanine. Pretty simple. Now, if I were to hybridize, that's a completely different story. Hybridization only occurs within correct connections. So for instance, cytosine would have to pair with what? Cytosine would have to pair with guanine. Adenine would have to pair with, let's say that this is another RNA. It would have to pair with uracil. Uracil have to pair with adenine, and then guanine has to pair with cytosine, right? So that's hybridization. And notice that the 5' end, or rather the 3' end, is connected to the tertiary alcohol, right? So let's write this again. So the third alcohol right here is going to be connected to this 5' phosphate, okay? So the connection still stands where we have a 5-carbon phosphate connecting to a 3' or 3-carbon alcohol, okay? So 5' phosphate connects to a 3' alcohol, 5' phosphate connects to a 3' alcohol, right? Very similar, is actually the same connection in DNA. So here we have RNA, right? We have RNA, it's very adorable, very cute. But notice that we have this guy right here, and he looks like he's having a great time, right? He looks like he's very happy. Well, this is just one strand of RNA. And you need to know that RNA is single-stranded. Single-stranded. Right? And it can actually connect to itself via these hybridizations. So notice that over here we have CCU, so we have CCCU, and then on the other side we will have GGGA. And that's just looking at this guy right there, his little leg. If we look at his other leg, we have hybridization occurring there. But again, remember that this is just one strand. So let's say that this is a 5' arbitrarily, randomly, and let's say that this is a 3' end. So again, we go from the 5' and we are connecting via the 3'. That's all I'm saying, right? And we can even loop right here. So what's happening here is that we have some unconventional hybridization. And it's really weird if you think about it, because look, if you zoom in right here, we have uracil, adenine, and cytosine. But then it's connecting to a cysteine, a guanine, and a uracil. And that's actually pretty weird. That's the only time you'll see it. And this mispairing, I guess you can call it, only occurs in RNA. Sometimes it happens in DNA, but it's always fixed, or most of the time it's fixed. In RNA, sometimes it's allowed to occur. And that could just be because a protein calls for it, or because it's a structural protein, or something like that. So RNA, sometimes it has unconventional, uncommon pairings. But that's just kind of like, you don't really need to know that. It doesn't really come up a lot. It's just kind of like for your knowledge, right? So as you can see here, RNA can have a specific 3D shape. So this is like a 3D model. And of course, RNA, it can have multiple functions. So RNA can be, it can have, structural. Let's see, let's write that better. So we have structural. It can be regulatory. It can also form or serve as a catalyst. And compared to DNA, DNA really is just more like a storage unit for information. Right? So that's just a storage information. Who cares? Not really that important. I'm just kidding, it is pretty important. But RNA can be structural, it can be regulatory, it can also be catalytic, right? And it can form proteins. But how do we make RNA from DNA? This occurs via transcription. So again, transcription is when DNA goes from, when the genetic material goes from DNA to RNA, right? So that's transcription. But how does that happen? Let's say that we have a pretty simple strand of DNA, that we need to make into RNA. Let's say that it's TAG, right? And it goes from the 5' end to the 3' end, okay? And now let's say that from the 3' end to the 5' end, we have the complementary strand. So complementary strand means that it is a strand of genetic material that can hybridize with the other strand. That's complementary. So the complementary of T would be A, right? So thymine goes with adenine. Adenine goes with thymine. Guanine goes with cysteine. Right. Pretty cool. So that is still DNA. Notice that it's DNA because we have adenine, right? And we also have thymine. So if it was RNA, thymine would be uracil. But now, let's transcribe this. So it's important to remember that whenever we are doing transcription, Rna polymerase likes to work in the 3' direction to the 5' direction. So let's write it over here, Rna polymerase. And Rna polymerase is the little enzyme. Notice that ACE means enzyme. That converts DNA into RNA. And it works from the 3' to the 5'. So therefore, I'm going to read this strand of DNA from the 3' going into the 5' direction. Okay? And it creates complementary strands in the 5' to the 3'. So you're probably saying, whoa, this is way too many numbers and primes and algebra. I don't know where you got algebra from, but hey, whatever. What I'm saying is Rna polymerase is going to read this strand of material if, and this is a big if it is going to read this strand of material, it's going to read it going this way. Okay? It just starts reading from the 3' to the 5'. And when I say it places something down from the 5' to the 3', I mean this. We're going to make a complementary strand of RNA to this strand right here. So we go first 5' to 3'. Okay, that's the basis I'm going to place down. So I'm going to read this strand from the 3' to the 5', and then I'm going to place down nucleotides, these little guys right here, from the 5' to the 3'. So let's do it. 3' adenine becomes a 5' uracil because we're making RNA. This thymine becomes an adenine, and the cysteine becomes a guanine. So notice something right here. This strand of RNA, it is complementary to this strand of DNA, right? Because they can pair up together. But notice that this RNA is very similar to this DNA. You can even say, hey, wait a minute. This strand that I just made is almost the same thing as this strand of DNA, except that this 5' thymine is a 5' uracil in RNA. And you know what? You'd be right. You would be correct. So what did we learn here? We learned that the template strand is typically the 3' to the 5' end. So the template strand, for our purposes is going to be 3'. Let's write over here 3' to 5'. And Rna polymerase works from the 3' to the 5'. Whenever I take a double-stranded DNA, I'm going to work from the 3' end to the 5' end. And I'm going to create a complementary RNA that is almost the exact copy of the coding strand. Now the coding strand is not transcribed. This guy right here is not transcribed. Let's write it better. Transcribed. Okay? But the sequence is equal to the RNA sequence, except that thymine is being replaced by uracil in RNA. The template strand right there, the template strand, is transcribed. So hopefully now you know that RNA polymerase works from the 3' end to the 5' end. It creates a complementary strand of RNA to the template strand of DNA, and it places down these nucleotides in the 5' to the 3' direction. Pretty good. So now you know. And this is actually very studied on the MCAT. So you will have questions on this. So how does RNA polymerase operate? Well, obviously it's an enzyme, and it's the enzyme that's responsible for creating RNA from DNA. Now notice that, again, I'm reading from the 3' end to the 5' end. If I keep repeating information, most likely it's pretty important. And it begins placing down these nucleotides from the 5' end to the 3' end. So notice that as RNA polymerase is operating, it's going to put this guy over there. Okay? So from the 5' end to the 3' end. It begins placing down these nucleotides by creating phosphodiester bonds. So of course RNA and DNA, so these guys, DNA, also RNA, they are connected via phosphodiester bonds. And essentially what that is that whenever we had this nice picture where we had the strands of DNA connecting to each other, they had the phosphate group and it was connected to this oxygen group, and then eventually it was connected to another phosphate group, and this was like the 5' carbon, this was the 3' carbon, 5' carbon. This right here is called a phosphodiester bond. That's all that is, okay? So let's clear this up. Pretty cool. And what happens here is that these free-roaming nucleotides that the RNA polymerase uses, these are called ribonucleoside triphosphates. So these are called ribonucleotide triphosphates. Okay, so of course I just butchered that spelling. Pretty good right there. So let's write this better. Triphosphates. You know, I was really bad at spelling at school, so whatever, STEM major, right? So right here, it's basically a free-roaming nucleotide. It has 3 phosphate groups on it, and that's pretty important because whenever RNA transcriptase, or excuse me, polymerase breaks one of these phosphate bonds, it releases a lot of energy, and that energy is going to be used by the enzyme to continue the reaction. So as it continues to break down these bonds, it uses that energy and it allows it to continue this reaction, right? Pretty good. And of course, it's going to be making phosphodiester bonds using this 3', you see 3' alcohol, right? And it's going to connect the 5' phosphate to the 3' alcohol. Okay, so that's pretty much it. So here we have a very cute and adorable RNA polymerase, and you can see that it's operating from the 3' to the 5'. It's creating a new RNA transcript from the 5' to the 3' direction. And this transcript is pretty much the equivalent of this guy, except that it has uracil. Okay? And it's complementary to the 3' strand of DNA. Okay? And notice that we have some ribonucleotide triphosphates, and they have to be free-roaming just around in the environment, and they're going to attach to the active site of the RNA polymerase, because typically enzymes, they have active sites where they do the operation. So here the active site, the operation of the active site would be creating RNA, right? And you need to remember that you don't just have one RNA polymerase working on one strand at one time. No. You have multiple, trillions of RNA polymerases working on the same strand of DNA. So these little circles that you see here, that you may see here, these right there, let's write it in a different color, these right here are going to be RNA polymerases. And they're working on these little strings, and these strings are complementary RNAs. So this black strand that you see over here, that's DNA, and these little strands that you see here are RNA. And they are being created by these little spheres, these little black spheres of RNA polymerase. There are multiple types of RNA. For instance, you have mRNA, which stands for messenger RNAs, and these codes for proteins. So for instance, the start codon, methionine, which is an amino acid, would be AUG, right? And so this AUG would actually code for a protein, right? It would serve as like the start codon for a protein. Okay, so this would be like a mRNA. It would go through a ribosome, and it would come out the ribosome as a fully formed protein. And later on in these lectures, we'll actually discover how these ribosomes create proteins. Now, speaking of ribosomes, we have rRNA. And this actually forms the core of the ribosome structure. So of course, ribosome, and I'll make kind of like a very simplified version of it. Right, so this is kind of like a ribosome. You have the big unit, and then you're going to have the small unit. And together, this makes up a ribosome. But the ribosome itself is created from rRNA. That's ribosomal RNA. So this is rRNA right there. We also have microRNAs, and this regulates gene expression. So for instance, microRNA, they are responsible for the activation or silencing of proteins. So for instance, if I have this protein, let's say, I don't know, kind of like this, right? So we have this protein right there, and let's say that I don't want to express it. Let's say that it's a cancerous protein, right? So it'll keep on replicating, right? And again, when we talk about cancer, we're going to be talking about these cancerous proteins, or proto-oncogens. Well, if I put a microRNA, it can interact with this protein, and it can silence it. It can actually silence the protein. So this is a silencer. And the way it does that is that it can cleave, it can cut this protein. And when it cuts the protein, it deactivates it. Or it can actually interfere with the connection between the protein and the ribosome, so that the ribosome cannot create the protein. And sometimes the microRNA can interact with the poly A tail. So later on, when we talk about the post-translational modifications of proteins, eukaryotic proteins typically have a end of the protein that has a bunch of adenines, right? And that helps to stabilize the protein within the cytoplasm. But the microRNA can actually remove this poly A tail, and it can destabilize the protein. And when the protein is destabilized, it'll decompose and not be activated. And so really, microRNAs regulate gene expression by silencing. Most of the time, they do silencing. Okay, so that's the thing you need to remember about microRNAs. And of course, we have transfer RNAs. So tRNAs are pretty fascinating. Again, we have the anticodon, AUG, and this is a start codon, that goes for methionine. So methionine is, of course, an amino acid, but it also represents the start amino acid. So whenever a protein is being made, to begin the process of creating a protein, you always have an AUG. And a tRNA, which is it's essentially like an adapter, okay? And we have like a whole slide about it, so it's pretty interesting. But the thing is, tRNA reads AUG, or it reads other codons, and it codes for those proteins. And those proteins are then going to create more proteins. Very fascinating. And of course, we have other non-coding RNAs. So let's clear the board really quickly. So one of the most prominent things that you have to remember for other non-coding RNAs would have to be the small nuclear RNA. And some people, they call them SNRPs, right? So what these guys do is that essentially they are going to interact with spliceosomes. So these are spliceosomes. Spliceosomes. And what these guys do is that they are going to help regulate or maintain transcription factors in the nucleus. So helps maintain transcription factors, in the nucleus. And we'll talk about transcription factors later. So sRNAs, they're also responsible for the maintenance of telomeres. So maintains telomeres. Of course, we have that right there. So they're pretty important to chromosomes. They're important for the nucleus, the maintenance of RNA, and yeah, they're very important, and they're always associated with these spliceosomes. In fact, whenever they are associated with spliceosomes, they're called SNRPs. So small nucleoribonucleic proteins. That's SNRPs, okay? And this is acid, and this is a protein. So whenever these guys are associated with spliceosomes, they become proteins. Small nucleic ribonucleic proteins. Very fun to say. And I guess small nuclear ribonucleic proteins. Within bacteria, notice that we have a RNA polymerase. So again, this is the same thing happening in bacteria. So this is bacteria. And we are going to start making RNA, right? So this is how bacteria can create RNA from DNA, right? So again, they're going to have an RNA polymerase, and they actually have a start site. So the start site is called a promoter, and the stop site is called the terminator. So the promoter site is where the RNA polymerase is going to bind to the DNA. And when it binds to the DNA, it's going to kind of split open the DNA and begin reading. So it reads again from the 3' to the 5'. That doesn't change, it's the same. But notice that we have this thing that kind of looks like a lemon. And unlike a lemon, this is actually called a sigma factor. So bacteria, one of the most important things to remember is that bacteria uses a sigma factor, uses the sigma factor. And so the sigma factor is more like a subunit of the RNA polymerase that recognizes the promoter region. So the sigma factor is going to recognize the promoter region, it reads it, and it's going to open up the RNA synthesis, right? So the RNA synthesis begins. And then afterwards, the sigma factor is going to be released, right? And once the sigma factor is released, the RNA polymerase is going to clamp down onto the DNA, and it's going to continue the creation of RNA, right? So RNA polymerase clamps down on the DNA, and it begins the creation of RNA, right? And so now we have the termination and release of both the polymerase and the completed RNA transcript. So whenever it's finished, the RNA polymerase is going to release the DNA. It goes back into the environment, and then the sigma factor is going to bind again with the RNA polymerase. Afterwards, let's say that it receives another signal from this DNA, a different DNA. So now the RNA polymerase is going to rebind with a different DNA, and it's going to read another promoter site, and then the sigma factor is going to recognize the promoter site and then release itself. When it releases, RNA polymerase continues to create RNA from the DNA. So again, the most important thing to remember is the sigma factor. If you have a question on an exam and it says the difference between bacteria and eukaryotic cells is that eukaryotic cells use a sigma factor, that would be false, because eukaryotic cells do not use sigma factors. Instead, bacterial cells use sigma factors. Very important. Within bacteria, so again, where we are talking about bacteria, we can actually create multiple, how should I say, genes from the same DNA. So notice that this is one strand, or two strands of DNA. But notice that I can have a polymerase, RNA polymerase, let's say over there, with this little sigma factor, and it's going to recognize this protein, and it's going to create gene A. So gene A, let's say it codes for, I don't know, a flagella. Gene A was created from the 3' end to the 5' end of, let's say strand A, okay, for strand A. Now gene B can be created from the same strands of DNA, except that it reads a different portion. So let's say instead of reading the bottom portion, let's read the top portion. And so from the top portion, if I read it from the 3' to the 5' on the B portion, I get gene B. And let's say that gene B codes for, let's say it codes for the cell wall, right? Let's say that it makes the cell wall. So notice that on the same strand of DNA, I made the cell wall over here, but on the bottom strand of the DNA, I made the flagella, right? So that's pretty weird, because normally you would say, well, you know, one DNA would code for one thing, or one strand would code for, I don't know, one thing, right? But for bacteria, two strands of DNA can code for different things. So gene A can code for, let's say, this portion would code for the flagella, and the other portion of the DNA would code for the cell wall. And so within these DNAs, you can code for multiple things. And that's how we can afford to have quote-unquote small or low amounts of DNA, but so many proteins or so many functions from that DNA, because different portions of that DNA can create different items. Before I begin my discussion with eukaryotic DNA and RNA transcriptase, right, or I guess polymerase, I need to talk about the different forms of RNA polymerase within eukaryotic organisms. So let's say that we have a chicken, and that chicken is being analyzed. Within that chicken, you're going to see different polymerases. So for eukaryotes, we will have 3 types of RNA polymerase. Okay. So we have RNA polymerase 1, so we have poly 1, poly 2, poly 3, and that's it. So what this codes for is that we are going to code for rRNA, so ribosomal RNA, so that makes the ribosomes. Right. Polymerase 2 is mainly for protein coding, so this is like mRNA. And then RNA polymerase 3 works for tRNA. So the way I remembered it is I say 1, 2, 3, and I say really mad teacher. So this is really mad, and this is teacher. Right. So again, this goes for ribosomal RNA, messenger RNA, and tRNA. And for eukaryotes, we don't have a sigma factor. Remember that bacteria do have sigma factors, but for eukaryotes, instead of having a sigma factor, we will use transcription factors. So transcription factors are used, not sigma factors. That's a really good F. Within the eukaryotic cell, when we begin the transcription from DNA into RNA, again, we are going to be using the transcription factors. So, in general, we're going to be using the general transcription factors. So we have the general transcription factors. And these general transcription factors, which are just going to be labeled as TFs, they are going to bind to a specific promoter region. And this specific promoter region is called a Tata box because it has a high amount of thymine and adenines. So it has TA, 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 and to ad nauseum. So it just has a lot of TAs, right? So what happens here is that we have the Tata binding protein. So this guy right here binds to the Tata box right there, and it allows these RNA polymerases to kind of bind to it. So these binding proteins assemble onto the Tata box, and notice that we keep getting more proteins, and these proteins eventually form a complex, right? So now we have this, I call it a Tata complex, and it allows the RNA polymerase to connect to it. Okay, it connects to it. So which one are we using? We're using the RNA polymerase 2. So if we're using RNA polymerase 2, what are we creating? We are creating mRNA. And if I'm creating mRNA, then what am I creating? I'm creating a protein. Another thing that's pretty important is that the general transcription factors are going to pull apart the DNA. So notice that right here we have this kind of circular thing occurring, and let's do that in a different color. Let's do that actually, you get to see my color wheel actually, it's pretty cool. So let's do it in this brilliant red, pretty cool. So right here you see this nice opening on the DNA, and that opening is facilitated by the general transcription factors. So of course they pull apart these DNAs, and that allows the RNA polymerase 2 to begin transcribing the DNA into RNA. Okay? And that's really all that you need to know about it. Yeah, of course, the RNA polymerase always works from 3 to 5, and it lays down from the 5' to the 3', right? So here you can see the Tata binding protein, and it's a subunit, okay? And notice that it binds, here's the binding protein by the way. This binding protein binds to the Tata box, so we have the TA section, and it's going to kind of manipulate the DNA. So it allows RNA polymerase 2 easier access, right? So it's easier to access. When RNA polymerase 2 binds to the DNA of a eukaryote, it's going to have a phosphorylated tail. So this tail that you see here is part of the RNA polymerase 2 complex. It will become phosphorylated, and whenever it is phosphorylated, meaning that it has a phosphate group attached to it, we have these RNA processing proteins. So these processing proteins are going to hitch a ride onto the RNA polymerase 2. And whenever this mRNA is being created, so again this is going to be the 5' end, and this right here is going to be the 3' end of the mRNA. As soon as it comes out of the RNA polymerase 2, these processing proteins attach themselves to it. So we have the capping factor. And the capping factor is actually going to make a 5' cap, okay, of guanine. So the 5', let's make some protein right here. So this is the 5', this is going to be the 3'. So whenever we have the capping protein, it's going to make some guanines. And then the polyadenylation factor is going to attach itself to the 3' end, and we're going to have a poly A tail. So poly means multiple, A means adenine, and we are going to add multiple adenines to the 3' end. So we're going to have this. And I'll show you how this looks like on an actual protein. So this is what it looks like. Notice that we have a guanine on the 5', that's called a 5' cap, and we also have a poly A tail on the 3' end of the RNA. And again, this was created by RNA polymerase 2. Now notice that the 5' cap is actually a methylated guanine. Methylated just means that we added a methyl group, which is a CH3 group. So notice that this this methyl group was attached to the guanine. And it looks like this. So we have our RNA right here. And then on the 5' cap of the RNA, so this whole guy right there is an RNA. On the 5' cap, we have a guanine. Okay, we have a guanine. And on this guanine is actually a methyl group, right? So again, this is our methyl group right here. This is our guanine, and this is called a 7-methylguanosine. You don't really need to know about 7-methylguanosine. You just have to know, okay, via the post-translational modification, modification. Really can't spell today. Anyways, so the post-translational modification is that for RNA, it's going to be added a 5' guanine cap, and on the 3' end, you're going to have a polyadenine tail. And that's all you need to remember. Within eukaryotes, we can actually modify the DNA itself. Well, what do you mean I can modify the DNA? Well, I can modify the DNA by removing some of it out, right? So this is called splicing. And splicing occurs in pre-mRNA. Well, what do you mean? What do you mean pre-mRNA? So when I say pre-mRNA, I mean this. So again, we have pre-mRNA. And sometimes this is called hnRNA, hnRNA. And what hnRNA is basically kind of like raw. It's raw RNA. And what I mean by raw RNA is that it has all the genes on it. And essentially, what we need to do to create good proteins is that we need to purify this raw RNA. Okay? So what are we going to do? Because right now this RNA is pretty raw. It has a lot of stuff that we don't need, so we need to remove some of the stuff that we don't need. And the way we do this is that we use spliceosomes. So we use spliceosomes. And these spliceosomes are going to remove part of the RNA, right? The part that they remove are called introns. So the thing in orange is called an intron. And these introns are not going to code for anything. As you can see here, they're called non-coding genes. And the way I remember this, of course, is via exons are going to express themselves. So exons express themselves, but introns are introverted, so they don't express themselves. Pretty cute. So now we're going to go into the process of how spliceosomes, which splice means to cut, how do they do this? Now, the way introns are removed from the gene is that spliceosomes, they react to very specific sequences. So hypothetically, let's say that this spliceosome is going to cut the bond between adenine and guanine, between the guanine and guanine, right? So let's say that this spliceosome is going to splice the bond between guanine and guanine. Now this changes for spliceosomes. Sometimes they receive a different signal to cut a different bond, but this spliceosome in particular has a signal that cuts the bond between guanine and guanine. Now notice that there's a bond between this guanine and this guanine right there. So what happens here is that a spliceosome is going to cut that bond, and it's going to cut another guanine-guanine bond right there, and it's going to remove this whole intron. So whenever this whole intron goes somewhere else, these exons are going to merge together, right? And these exons, they create a new section. So exon 1 connects to exon 2, and it creates a guanine-guanine bond. So it's kind of weird if you think about it, but it doesn't get processed by the spliceosome, if that's what you're thinking. It has a signal that stops the splicing action, right? So again, just to recap, the intron is removed via a very, very specific bond, okay? So it's not always going to be guanine-guanine. It can change for different spliceosomes. And of course, whenever we're talking about spliceosomes, we're going to be talking about SN RNAs, because SN RNAs are always associated with spliceosomes. Okay. And the way that spliceosomes do this operation is that they work with a Lariat structure. So this is a Lariat structure. And Lariat just means, so you ever seen like those Western movies where they're harassing these cows and they're cowboys, they have the little rope that they swing around? That's called a lariat, right? So I don't know why I was calling it Lariat. Weird. But this is a Lariat structure. So a lariat is kind of like a lasso. Okay, so not Lariat. Who do I think I am? I'm not British, gosh. So this Lariat structure is very specific. So you can see that this is a pre-mRNA, also called HNRNA. And let's write that better, HNRNA. And the intron sequence is going to be pinched off from the exons, and as it is pinched off, it forms a Lariat structure, and it's going to be removed. And notice that exon 1 and exon 2 eventually form into one exon. So exon 1 and 2 connect. So now this Lariat structure is going to be degraded within the nucleus, because everything that we see here is pre-mRNA. So we're not even mRNA yet. So this occurs in the nucleus. So if you see a question on the test that says, oh, this occurs in cytoplasm, that's wrong, because the cytoplasm is outside of the nucleus, and this whole operation occurs within the nucleus. So let's see if we can actually see this process in action.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
I really do hope that you're enjoying the movies that I'm putting into the lectures. I know that whenever people are lecturing on cell biology, it tends to be very PowerPoint-like, and I don't really like teaching on slides. I like learning visual, so hopefully you enjoy the movies. And if you do, you can leave a comment down below, and you can make my day. It's pretty nice. So now, we're still talking about the eukaryotic organisms, so we have the eukaryotes. And you're probably wondering, well, hey, wait a minute. I have DNA, and so you're telling me that this DNA codes for multiple things, but I can't see it. How does that happen? Well, the way that your DNA codes for your eyes or for your skin or for your fingernails is that we can splice out some of the introns and selectively choose which exons we want to pick or we want to express. So again, this pre-mRNA is going to have the exons and also some introns. But what happens if I get rid of the introns? So if I get rid of all the introns, I'm going to have all the exons. So I have exon 1, exon 2, exon 3. And let's say that I need all 3 of them to create I's. Okay? But what if I ignore exon 1? So what if I just get rid of that? And I just want exon 2 and 3. What do I get? Well, let's say that if you get 2 and 3, you'll get, I don't know, like some eyelashes. And for this one, if I ignore exon 2 and I just put exon 1 and exon 3, then let's say that I get eyelids. And so notice that we code for different parts of our body using the same strand of pre-mRNA, but we chose which exons to express. So the combination of exon 1, 2, and 3 gave us eyes. The combination of exon 1 and 3 gave us eyelids. The combination of exon 2 and 3 gave us eyelashes. And that's kind of like the whole basis of epigenetics. So epigenetics kind of rides on this concept. And so if you ever asked yourself, well, how do I get different body parts and how do I code for different things using the same DNA? That's how we do it. We do that via alternative splicing. So this whole process is called alternative splicing. Okay? And that is how we get different body parts or different proteins from the same pre-mRNA, also called hnRNA. So do you remember when I was talking about general transcription factors and how they allow eukaryotic organisms to transcribe RNA from DNA? Well, you should also know that for eukaryotic transcription factors, there's also regulators. So again, we're going to have transcription regulators. Transcription regulators. And these regulators, and that's an A, these regulators actually act from a distance. So they act from a distance. So notice that we have Rna polymerase 2. Okay, and it's going from the 3' end to the 5' end. And what it does is that these activator proteins, these transcription regulators, are going to act on a mediator protein. So let's kind of step back for a bit. Remember when we were talking about general transcription factors and how they accumulate proteins and these proteins form a complex? Well, how does that complex begin? Right? Did you ever question how that complex starts activating and separating the DNA so that RNA polymerase 2 can activate? Well, it activates using an activator protein. So here we go. This is a eukaryotic activator protein, and it has an enhancer, which is a binding site for this activating protein. So now, when this DNA has its activator protein, it's going to activate a mediator protein. This mediator protein connects to the transcription complex, which is this guy, and then that transcription complex just separates the DNA, and it allows the RNA polymerase 2 to begin working and creating RNA. So let's, I know it's a lot, but let's recap. Okay, so the activator protein activates the mediator protein. The mediator protein is working on the transcription factors, and this is what activates the transcription factors. When activated, the transcription factors allow the RNA polymerase 2 to begin working. And that's all you need to know. Another thing that you need to remember is that for eukaryotic transcription regulators, they can actually regulate histone modifications. So we can say that transcription regulators regulate histone modification. So what happens if I put histone acetyltransferase? What do you think will happen? Well, if the transcription regulator binds, so this guy, if we bind to a histone acetyltransferase, what we're doing is that we are transferring acetyl groups. Now remember when I said acetyl groups, they actually up-regulate DNA expression. So again, acetyl groups increase DNA expression. And so it goes to show that if I have histone acetyltransferase, I'm going to have more RNA. So I have more RNA. Okay, but transcription regulators can also bind to the chromatin remodeling complex. And the chromatin remodeling complex is going to convert this heterochromatin into euchromatin. So remember that euchromatin, when I have euchromatin, I can actually express the genetic material. But when it's heterochromatin, it's more compact, and the DNA cannot express its genetic material. And so what I'm trying to say is, despite whichever pathway you go, you will always have more RNA for this for these routes. Of course, we can have the reverse reaction, where we can have some methyltransferase, and I could downregulate the DNA expression. Or I could just make more heterochromatin. So it really depends on what we need. So the transcription regulator can upregulate, and it can also downregulate. In this picture right here, this is only showing the upregulation. And so whenever we have a eukaryotic cell, we have so much stuff going on. We have a combination of transcription regulators. And whenever we have a combination, we call this combinatorial. Combinatorial control. So notice that we have the chromatin remodeling complex. We also have the histone modifying enzyme. We have the Tata box. We have transcription regulators, general transcription factors, but we all covered it. We covered it all. And so that's how your DNA is being converted into RNA, and that's how it regulates itself. That's how it upregulates, downregulates. It does everything. And it's very amazing. And so within you have a complex system of enzymes and DNA and RNA, and that's what makes you, you. And that's why you're special. Within eukaryotic organisms, the alternative splicing and the combinatorial control of DNA into RNA can create different cells. For instance, we can convert a liver cell into a neuronal cell. And really, we can do that in the lab. We can do this artificially, right? So, for instance, I can introduce a specific transcription regulator. So, if I were a scientist and I wanted to do transcription regulation, I could put a needle or something, and I could put some regulators. So if I put a regulator into a liver cell, hypothetically, just for this example, I can convert the liver cell into a neuron. And that's really, really impressive, because we can do this artificially. Now, scientists are kind of messed up, and a lot of weird people are scientists, you know. Like, and so what happens here is that they're going to be using combinatorial control, again, combinatorial control, to create different cell types and different organs. So here's this innocent little fly called Drosophila melanogaster. It's a common fruit fly, and they are going to take some factors from its eye, and they're going to implant it into a different part of the leg. And so whenever they put this transcription regulator into the leg, they are going to express a different cell. And so for this cell specifically, they created an eye. So this is an eye structure formed on the leg, and that was artificially expressed using precursor cells. And so it's very disgusting. It's kind of freaky. But I don't think this eye works because it's not really connected to nerves. It's just the cells themselves were expressed into the eye cell, right? So I don't know if there's nerves connecting to it. So I don't think it can see. Or can it? And now we will watch a short video about everything that I just covered, and that will encompass the transcription between DNA into RNA in eukaryotes. I hope you enjoy the video.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme Rna Polymerase. It maneuvers the Rna polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The Rna polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
That was a pretty cool movie, wasn't it? I thought it was pretty cool. Anyways, I hope that you understand how RNA is being transcribed from DNA, and that how it uses different factors and mediator proteins and activator proteins to create RNA. And the main mechanism is RNA polymerase II for eukaryotic cells. So hopefully you understand that, and that this video helped you in studying for your exams. And yeah, so thank you so much for watching, and I hope that you have a great day. And remember, as always, that I love you. You're so amazing. All right, goodbye, and thank you.